Hi, I am Jennifer Hackney Price, and today I'm going to show you how to make embryo collection plates. These are the same ones that I use in my lab whenever we need to collect embryos or collect staged larvae. So the end product is going to look something like this. It's a standard 35 by 10 millimeter plate. And when we get done, we'll allow the flies to lay some eggs on there. We could incubate it for however long we need to and get the stages that we would like. And I'll show you how to set all that up here in a moment. First thing we need to do though is cook the agar that's going to go into these plates. So I like to use this grape agar from Genesee Scientific. You can get it from a bunch of other places as well, but this is what I found to be very convenient. One of these little packs uh, makes 500 milliliters of grape juice agar, and that will make approximately 100 plates worth of embryo collection plates. So I'm going to start by adding the water. And again, it's 500 mils for every packet of food that we're going to use. Get that started spinning. All right, so I just turned up the stir bar and turned up the heat a bit. I'm just going to open up the package. Again, this is super simple. This is one reason I like it. I'm just going to take that and pour that in. This, by the way, is one of the best smelling things that you're ever going to be able to do in a lab, because you can really smell that grape juice coming out of there. So we're going to adjust the stir bar just to make sure that it's spinning properly. Once it's spinning and heating up, then we're just going to cover this up. And we're going to let this cook. It's going to take about 15 minutes to get to the consistency that we want it to. One word of warning is that this stuff boils over very easily. So you want to keep a close eye on it, because as soon as this stuff starts getting hot, it will start to boil over. So just be aware and pay attention as you're cooking this. So we're going to heat this to boiling. Again, it'll take about 10 minutes. I like using the hot plate. If you have microwave available, you can also melt the agar in there to prepare the plates instead of using the hot plate. So the food's been cooking for about 15 minutes. It started to boil, so I immediately turned the heat off and took this off of the hot plate. Again, make sure you're using your hot oven mitts just to make sure you don't get burned as you're doing this. Now we're ready to pour the plates. You don't have to let it cool down. In fact, if you let it cool down too much, it will solidify before you get a chance to pour all your plates. So you want to do the next steps fairly quickly as soon as it starts to boil. So here's what we're going to use the plates for, is to set them into these embryo collection cages. So the cages are designed to fit one of these agarose plates in the bottom. I tend to use the little embryo collection cages. It's just convenient for my experiments, but they do make them in a larger size if you need a lot of embryos or are working with a larger scale experiment. Today we're going to stick with the small plates. And again, I use these 35 by 10 millimeter plates. When you're getting plates for this, just remember, do not use any of the ones that have the lips on them, because they will not fit in the cages properly. You can just pour the plates using the pour spout on the beaker, or you can use a serological pipette if you want to be a little bit more accurate. I think lots of teaching labs have this nice little green pipetteman that will allow you to do this. So to fill up one of these plates, it's going to take approximately 5 milliliters. All I'm doing is filling that up and then just putting the lid on. A couple of them here. So you might get some air bubbles in here. That is not a big deal. Flies don't really care if there's an air bubble there or not. But once you have those poured, you want to let them set. And you'll know that they're set when the solution inside there starts to look cloudy. It doesn't take very long. Usually it takes about 5 to 10 minutes for them to set, at which point you can put them in the refrigerator for longer term storage. I like to store them upside down in a sealed Tupperware container. This is so that you don't get a lot of moisture accumulating on the plates, and when you put your flies in there, they don't get stuck to the agar in the plate. Once we have our embryo collection plates, now we could actually use them to collect some embryos. So again, this is my embryo collection cage. You get them online fairly easily. As you can see, there's a little hole in the bottom of this where you put your newly made embryo collection plate. And the rest is going to be a container where you're actually going to store your flies. So the first thing you want to do is take the lid off your plate and set it into this round ring. Now, some plates fit a little more tightly than others. I just happened to get one that fits fairly snugly in here, so I'm going to push it down with the container. But you can see it makes a nice little bottom for our embryo collection lid. So once our plate is ready to go, now you're ready to transfer your flies in there. And the number of flies you put in there really depends on what you're trying to accomplish. You could track egg lay for a single female, or you could have a whole population of flies in there. So it really depends on what you're trying to accomplish. Our standard is approximately 30 for the things that we want to do. So just like when you're transferring flies to a food vial, the secret is going to be tapping. So you're going to keep tapping. And because I'm going into a container of a different size, I want to make sure I use a funnel because this will help us to minimize how many flies are able to escape. So I'm going to tap down, take the lid off, invert, and then continue with my tapping. That's down. I'm going to seal it with our embryo collection plate. And that is now ready to go. And you could incubate that however long you want. In our lab, we typically do 4 hours of collection times because we want pretty precisely staged embryos. But you can do much longer than that or shorter than that, depending on what you prefer. One thing to note, especially out here in the desert in Phoenix, where we are, these things have a tendency to dry out if you leave them at room temperature. So one thing that you can do to avoid that is set them in a small Tupperware container with maybe a centimeter's worth of water on the bottom just to increase that local moisture content. But once this is done and you've allowed them to incubate as long as you want, then you're going to do the exact opposite to get your flies back in the container. Again, tap, tap, tap. Flip it over. And seal with the plug. Now you have a plate that you're ready to examine under the microscope or do whatever it is that you want to do with it. So we have switched locations. We are now in one of our teaching labs so that we could look at what you might expect to see under one of our embryo collection plates. So on this embryo collection plate, there's a number of eggs. And I'm going to put one into focus here. There we go. So you can see an egg right here. There's the bulk of the egg over here. There's 2 little kind of appendages sticking out. These are called dorsal appendages. These are what the embryo uses for gas exchange. So flies are going to lay eggs on things that are very kind of wet, rotting fruit. But the embryos still need oxygen. So these dorsal appendages serve as gas exchange, allowing the embryo to still get air even if it is partially submerged. So this is what we're looking for. This is a wild type egg. There's lots of different mutations that could affect what these dorsal appendages look like. There's some that can also influence how many they are. When the larva first hatches, it will be the same size as that egg. So don't expect any huge larvae. They will look almost exactly like that, except for they will have mouth hooks and no dorsal appendages.